These videos are offered on a pay-what-you-like basis. You can pay for the use of the videos by making a monthly pledge at my Patreon page. Or you can make a one-time payment by using the PayPal donate button on my website. Thank you. What's the name of this amino acid?
That is that threonine?
Like this?
A serine.
Yeah, threonine would look like this. What's the name of this amino acid?
Leucine.
Like this?
Valine.
Like this?
Yes.
Okay. How is leucine different?
Leucine has one more CH2 group. Has a CH2 group in between. The alpha-keratin and the nitrogen.
Oops. Okay. Well, no. Actually, okay. Yeah, that's right. So this would be leucine. It also has a V structure at the end, but with one extra CH2 group. Okay, so those have to be memorized. It's important to keep working on those until you have those down. What amino acid is this?
Is that aspartate?
Right, you can see I'm using bond line notation here to make it take less time to write these out. And what amino acid is this? So that's good. This is aspartate. What's this?
That is lysine.
That's right. Okay, again, you just have to keep working on those until those are easy for you. Okay, so I want to move on to the next topic here. What role do so what type of molecule is serine? What type of molecule is serine, valine, aspartate, and lysine? What group of molecules do serine, valine, aspartate, and lysine fall into?
What group of molecules?
I'm going for a pretty simple idea here.
Amino acids.
That's right. They're all amino acids. Why are amino acids important? What role do amino acids play in the body?
They make up proteins.
Yeah, they are protein monomers. So our next step now is to see how to put these together to form proteins. So the way this works is that we're going to connect this carboxy to this nitrogen. Maybe I'll show it like this. And this is something I'm pretty sure you'll have to do on the test. You're going to be expected to draw short proteins, short peptides. So the next thing is to make sure we know how to draw these short peptides. Okay, so what I've done here now is I've combined serine and valine. So you should study how I did this because this is something you'll need to do on the test, I'm pretty sure. The basic idea here is, do you recognize this functional group from your organic chemistry class? Do you know what the name of this type of functional group is with a carbonyl connected to this nitrogen? Do you remember that? So I was asking you, do you remember what type of functional group this is with a carbonyl connected to a nitrogen?
Is that a nitro group?
Actually, no. Nitro groups would look like this. Nitro groups won't be too important for your biochemistry course. So this group is called an amide. That's an important term to know for your course. This is an amide, a carbonyl connected to a nitrogen. Anyway, notice that the way we construct this is, notice that this oxygen drops off. This oxygen drops off. And the nitrogen changes to neutral. The nitrogen changes to neutral. How many bonds does a neutral nitrogen form?
3.
So how do I know? So how many hydrogens does this nitrogen need?
2.
Like this?
No, 1, I'm sorry.
How do you know 1?
Because now it has 3 bonds.
Yes. Okay. So I'm just trying to go over with you how to draw this correct bond. So notice the pattern here. The pattern here is nitrogen, alpha carbon, carboxycarbon. This is still called a carboxycarbon, even though it's not a carboxylic acid anymore. And then again, nitrogen, alpha carbon, carboxycarbon. That's a very important slogan to memorize for proteins. So peptide structure is nitrogen, alpha carbon, carboxycarbon. Nitrogen, alpha carbon, carboxycarbon. Nitrogen, alpha carbon, carboxycarbon. Nitrogen, alpha carbon, carboxycarbon. So we want to memorize that slogan. That's the way to make sure you've drawn the peptide correctly. Nitrogen, alpha carbon, carboxycarbon. Nitrogen, alpha carbon, carboxycarbon. Nitrogen, alpha carbon, carboxycarbon. Okay, so we can see we got that correct here. Nitrogen, alpha carbon, carboxycarbon. Nitrogen, alpha carbon, carboxycarbon. All right. Let me point something out. Is that making sense so far?
Yes.
Let me point something else out to you. Notice that in order to draw this smoothly, this side chain is pointing up, but then this side chain is pointing down. Do you see the difference? Notice how here the bond from the nitrogen goes up. And here the carbonyl is pointing down, but here the carbonyl is up. So this is the way to draw peptides. The side chains, when we're drawing these out, they're drawn, if you're using this type of bond line notation, the side chains alternate which side they're going to be pointing, up, down, or up, down. That's why up here, I don't know if you noticed, but I drew the valine amino acid kind of upside down relative to the serine because I knew I wanted to connect them. And then I drew the aspartate kind of upside down from the valine. For example, here the side chain is pointing down, but here the side chain is pointing up because I knew I wanted to connect those. And then I drew the lysine upside down from the aspartate because I knew I was going to want to connect those. Okay. Make sense so far?
Yes.
Let's turn on your screen. Okay, so on the left side of your screen, I'd like you to draw a dipeptide with serine connected to valine. Try doing this. And all these questions, we want to be practicing the amino acids, so try to do this without looking up the amino acids in your notes. Try serine connected to valine.
Serine connected to valine. Okay.
Okay, very good. And we can label that left-hand amino acid as SCR for serine, and the right one is VAL for valine, or you can write out the word if you want. Now, let's connect the valine to aspartate. Let's extend this chain by connecting the valine to aspartate.
Okay.
That's good. That's good that you took your time and kind of worked that out. So one thing you worked out is that one of the oxygen gets dropped out, and oxygen gets dropped out. Do you remember what the slogan is? Remember the slogan for these chains is nitrogen, alpha carbon, carboxy carbon. Trace that out with your cursor. Trace out the nitrogen, alpha carbon, carboxy carbon pattern.
Nitrogen, alpha carbon, carboxy carbon. Nitrogen, alpha carbon, carboxy carbon. Nitrogen, alpha carbon, carboxy.
Notice that one of the oxygens has to get dropped in order for that pattern to work. Okay, good. So now let's connect lysine to that chain.
Okay.
Good. That one has a long chain. We can label that as lysine. So what type of molecule do we have written on your screen?
That is a peptide.
Good. And what type of molecule? What type of molecule is peptides?
They are nitrogen, or they're amino acids.
That's not correct. Peptides are not amino acids.
They are.
Maybe that question didn't quite make sense. So what I was going is peptides are proteins. Peptides are just short proteins. Peptides are just short. Do you see why it's wrong to say they're amino acids? This is not an amino acid. It was made out of amino acids, but it's like suppose I don't know. Suppose you go up to a brick house. What is the brick house? No, that analogy is not going to work. All right, well, forget about the analogies. But anyway, it's not an amino acid. It's a protein that was constructed by connecting amino acids, but it's not an amino acid anymore. Okay, so what we're learning here now is how to make proteins. Is this a polymer?
Yes.
Yep, because it has these repeating subunits of the amino acids. Very good. Okay, point to the bond that connects the serine and the valine. Point to the bond that connects the serine and the valine. That's right. What type of functional group do we have around that bond?
An amide.
An amide. Now, there's a special name. It could be called an amide bond then, but there's actually a special name for the bond that connects the amino acids in a peptide. There's a special name for the bond that connects the amino acids in a peptide, and that's likely to be tested. Do you happen to remember that from class? You and I haven't talked about that, but do you remember from class, what's the name of the bond that connects amino acids in a peptide?
Polypeptide bond.
That is the right answer, yes. So you should label that's a peptide bond. That's an important term in the course. The way to do that is put an arrow pointing to the bond. So what you have right there isn't quite good because it makes it look like the nitrogen is the bond. Make like a regular arrow that points to the bond. The peptide bond. Good. Point to the bond to the left of that arrow. To the left.
Oh.
No, to the left of the arrow.
Oh, the left of the arrow. This bond?
Right to the... It's too bad I can't point with my finger. All I wanted you to do is go to the bond one step to the left. Yes. I just wanted to point out, would you agree that is not a peptide bond?
Yes.
Okay. Now point to the bond that's to the right of your arrow.
This one?
Yeah. I just wanted to point out, is that a peptide bond?
No.
No. So that's something that you likely need to do in the test. You have to be able to identify the peptide bonds. The peptide bonds are between the carboxy carbon and the nitrogen. Nitrogen, alpha carbon, carboxy carbon, peptide bond. Nitrogen, alpha carbon, carboxy carbon, peptide bond. nitrogen, alpha carbon, carboxycarbon, peptide bond is the pattern. So label all the peptide bonds that we have so far. You can just
star them.
That's fine. So how many are there?
3.
That's right. Okay, good. So that's important. Let's see. Okay. Okay, so now let's connect what we have so far. Let's connect what we have so far to isoleucine on the right. You can scroll to the right if you need to.
Okay.
Excellent. You can label that as ILE for isoleucine. That's very good. That's a tricky side chain, so that's good that you got that correct. Okay, doing good. Now let's connect proline. Let's add proline to the chain.
Okay. Okay. Is that right?
Okay, that's good that you were thinking about that nitrogen. Now, it turns out that amide nitrogens are never charged. Amide nitrogens are always neutral. Amide nitrogens are always neutral. They're not like amines. Amide nitrogens are not basic. Amide nitrogens are not basic, so they never pick up a proton. They're always neutral. So what should be the charge on that nitrogen?
That should be neutral. So
Because of what we just said. So how many hydrogens does it need?
none.
Okay, so I could see you were thinking about that, but I hadn't quite mentioned before, amide nitrogens are always neutral. Amide nitrogens are neutral. They're not like amines. So proline's a little bit tricky. Now there's one little detail here, which is if you're using bond line notation, you don't have to draw the hydrogens. But if you actually draw the letter C, then you do have to show the hydrogens. So where you put your alpha carbon there, you either have to connect the hydrogen, or better yet, just use bond line notation and don't draw the letter C there, if you see what I'm saying. You can't write the letter C unless you write its extra hydrogen. So the best thing is just to stick with the bond line notation, so you don't need to include the hydrogen. Okay, outstanding. Let's add histidine. we can draw the major form at physiological pH.
Okay.
Excellent. Histidine is hard for me to draw, but that's a good picture. Excellent. We can label that as histidine. What is the pKa for the carboxyl end on the main chain? What would you say is the pKa for that carboxyl end?
About 2.
Good. Now, actually, it turns out that it's only 2 for free amino acids. In a peptide, it changes to 3. The carboxy end actually has a pKa of 3 in a peptide. In a free amino acid, it would be 2, but here it's 3. In fact, if you still have your textbook with you, let's take a look at page 41 again.
Okay.
Do you see the terminal carboxyl group there?
Yes.
And what's its pKa?
3.1.
So we can approximate that to 3. So earlier we were using 2 because we were working with free amino acids. But in a peptide, the pKa changes to 3. Usually you're going to be working with peptides from now on, so that's probably the better number to memorize. If you're only going to memorize one of those, you should probably start using 3 instead, because we're going to be working with peptides mainly now and not separate amino acids. So that pKa would be 3. Also, the terminal amino group changes a little as well. What's the pKa for the terminal amino group?
8.
If you remember, earlier we were using 9. It would be 9 in a free amino acid, but the pKa's get a little closer to each other in a peptide. So like I said, probably it would be better from now on to use those numbers 3 and 8 instead of 2 and 9, because you're usually going to be working with peptides now, not amino acids. I don't know if your instructor is going to give you a problem that tests the difference between that or not, but it's covered in the textbook. Okay, so what is the net charge of this compound at physiological pH? Let's determine the net charge of this compound at physiological pH.
Of the entire molecule?
That's right.
Okay.
This is a pretty typical type of question for tests. Your instructor did a couple examples like this in lecture, so we want to make sure we know how to do this. You'll have to zoom out or scroll.
Okay, so no positive, negative, that's neutral. Positive, negative, so it's neutral.
That was fast? You said the net charge was neutral overall?
Yeah.
All right, let's see. So we got +1, -1. Okay, very good. So remember that the textbook already has all the groups written in their main form at physiological pH. All right, so now let's figure out the net charge at a pH of 5. Let's figure out what the net charge would be at a pH of 5.
So get a positive charge. The alpha amino get a positive charge.
Let's review something we were talking about earlier. Remember that only the amines can be protonated. The amide nitrogens are not acidic or basic. They never get protonated. Only the free amines can be protonated or deprotonated. The amides don't get protonated or deprotonated.
I think we're good.
Everything's exactly the same as before?
Yes.
Let's see. So the best way to do this type of problem is to write down all the pKa's next to the functional groups. So let's go through and write down all the pKa's close to what they're referring to. Good. Except, do you remember a moment ago we were saying that 9 is the pKa in a free amino acid? It would be better now to stick to what that table in your book says and use 8 for a peptide. And let's go back to that side. 2 was the pKa that we were using previously for free amino acids. But remember a moment ago we were talking about how the table in your book has a number of 3, which applies to peptides. So I guess it's a little confusing that we're shifting from 9 and 2 to 8 and 3, but that's because we were working with free amino acids before, and now we're working with peptides. From now on, you're mainly going to be working with peptides. That's what you're probably going to be tested on. So if you only want to memorize one set of numbers, it should probably be the 8 and 3 pair instead of the 9 and the 2. We'll have to break that habit a little bit. Okay? So let's write down any other pKa's that apply here. Ballpark numbers.
Okay.
I think you left out the lysine.
Oh. Lysine is 10. Or 11. Okay, so serine, which is 8. All right.
So what's the net charge?
Net charge is positive. Positive 1.
Okay. Now, that's not the answer that you were thinking maybe a minute or two ago. So it turned out that the answer here changed when we changed from the pH of 7.4 to 5. So these have to be done pretty carefully on paper. A couple other points to make here. I noticed that you wrote down that serine has a pKa of 8, but it's probably better not to say that's the serine, because it's not the serine side chain. So for that 8, the best thing to write there for the 8, that is what we can call the N-terminus. So instead, where you have written serine 8, it would be better to write that as N-terminus. And then for the histidine, you wrote down histidine 6, which is correct because that's the side chain, but we should also write down the pKa for the C-terminus, the carboxyl terminus. What's the pKa for the carboxyl terminus?
I'm sorry?
Look at what you wrote down for histidine. You can see that you wrote down histidine 6. Now, that refers to the side chain. That's fine, but you left something out. You left out the pKa for the C-terminus. Do you see what I mean by C-terminus? I haven't used that term before. Can you point to the C-terminus?
The C-terminus, this?
Okay, yeah, I haven't quite used that terminology before. The C-terminus is the far right side, the carboxy, and there on the far right side. What's its pKa?
3.
But notice you left that out of your little chart that you wrote up above, so you might have made a mistake. So you should add that to the bottom. You can label that as C-terminus. Does that change the answer?
No.
No. Yeah. So you got it correct, but to be systematic, we have to include that in there. Okay. So this is the kind of term notation that would be best for this type of problem. Very good. Okay. Now, all right, so let's try another exercise on this. Like I said, this is pretty sure to be a problem on the exam. So now let's try working out the net charge at a pH of 10. Let's work out the net charge at a pH of 10.
So, good. Okay, and that's going to be neutral. -1.
That's right. Okay. All right, so that's an important type of question to practice. Like I said, your instructor did some examples like that in class. That's a very typical type of exam problem. I myself tend to make lots of careless mistakes on those problems, but it looks like you've got a pretty good method for working those out. Let's label, again, all the peptide bonds in the peptide you have so far. Let's label all the peptide bonds. Okay, good. Good. Okay, I'll go back to my screen. So let's see here. What type of molecule is this?
Polypeptide.
Good. In particular, this would be called a tripeptide. Why is it logical to call this a tripeptide?
It's made up of 3 monomers.
Oh, hold it for a second. Actually, I guess I messed that up, didn't I? I guess it would be called a tetrapeptide, actually. All right, so much for that. Well, what do you think a dipeptide is?
Made up of 2 monomers.
2 amino acids. And what's a tripeptide?
3 amino acids.
Good. What's a hexapeptide?
6.
That's right. Hexa is 6. What's a pentapeptide?
5.
That's right. You know what a tetrapeptide is?
4.
That is right. Okay. All right. Now, it's important to notice that these amino acids have 2 different ends. Notice these 2 ends are not equivalent. There's a nitrogen on the left end and an oxygen on the right end. The 2 ends are not equivalent to each other. They're different from each other, so we need 2 different names for the 2 ends. This end, I've already started using this term, this end is called the N-terminus. You could just call this nitrogen the N-terminus, or you could call this whole amino acid the N-terminus, depending on the context. And this end is called the C-terminus. You could also call this the amino terminus. And this is the carboxy terminus. Okay, do you know why this is called a terminus? You know what term means? Term means end. So if someone is terminal, they're at the end of their life. Or the terminator ends other people's lives. Or a term paper is delivered at the, or a term paper could be the end of your life. All right, so anyway, term means end. So these are the 2 ends of the peptide, the carboxy terminus and the amino terminus. Those are important terms. What's the pKa for the amino terminus? Ballpark? Okay, let's see. So we were explaining that term means end. So the N-terminus and the C-terminus are the 2 ends of the peptides. These are important terms. N-terminus, amino terminus. I was asking you a question. I was asking, what's the ballpark pKa for the N-terminus of a peptide?
3.
Yeah, and for the C-terminus of a peptide?
The C-terminus of the peptide. 8.
That's right. Earlier we were using 2 and 9, but those are for free amino acids. For a peptide, 3 and 8 are better numbers. Now, peptides are usually written with the N-terminus on the left and the C-terminus on the right. They're not required to be written this way. If you're writing the structure, you don't have to write it this way, but this is the most usual way. So this is usually the best way to write the bond line notation from the N-terminus to the C-terminus. Okay? What do we call this bond?
Peptide bond.
Let's investigate that peptide bond a little bit more. Do you remember the idea of resonance from organic chemistry?
Yes.
Yes, okay. I hope it brings back good memories. Well, I won't torture you by asking you to draw the resonance structure, but you might remember that we used electron pushing arrows like this to draw resonance structures. So let's look at the other resonance structure for that peptide bond. I didn't manage my space here very well. Maybe I'll erase this. If you remember the rules for electron pushing arrows, since this nitrogen is at the beginning of the arrows, it loses electrons and becomes positive, and this oxygen becomes negative. So this is the other resonance structure for this peptide bond. I could also draw new resonance structures for all the other peptide bonds, but let's just focus on this one right now. What do we learn from this resonance structure? What do we learn from this resonance structure? What can we learn from this resonance structure? Well, first of all, notice that this resonance structure has a double bond. That shows us that this bond has double bond character. It's kind of a mixture of a double bond and a single bond. Now, why is that interesting to us? So here's a little bit more stuff from organic chemistry. Do you remember from organic chemistry, is there free rotation around single bonds, or are single bonds rigid? Is there free rotation around a single bond, or is a single bond rigid?
There's free rotation around a single bond.
How about around a double bond?
No.
It's rigid.
It's rigid.
It turns out that because of this double bond character, peptide bonds are rigid. There's no free rotation. There's no rotation around peptide bonds because of this resonance structure that gives it double bond character. Peptide bonds are rigid, no free rotation. Another important point, do you remember the geometries from organic chemistry? Tetrahedral, trigonal planar? Normally, do you remember what type of geometry do we have around a double bond? Around a double bond, we would have trigonal planar geometry rather than, say, tetrahedral geometry. So it turns out, because of this double bond character, that peptide bonds are planar. Peptide bonds are planar, as all double bonds are planar. You wouldn't see that from this resonance structure. Do you remember what the word planar means?
Flat.
So it just means it's flat. It's a flat plane. Can the flat plane rotate? Well, that was actually that was a dumb question. Forget it. All right. It's my point though is that it's planar. It's flat. So the things that are in the plane. So what we have here is that this hydrogen
Okay.
So here's the double-bonded atoms. This hydrogen, this alpha carbon, this nitrogen, this carboxy carbon, this oxygen, and this alpha carbon, they're all in the same plane. That's what we mean when we say the peptide bond is planar. These 1, 2, 3, 4, 5, 6 atoms are all in the same plane. This is a planar bond. Okay, there's no rotation around this bond. So you can imagine this all as this circle, or as a kind of a flat plane. One other point that's important, do you remember what's the definition of a base?
Something that accepts protons and donates electrons.
Excellent. So here we have a amino group. Whoops, I didn't mean to do that. This is an amino group, and here we have an amide group. Now remember, we've seen, are amino groups acidic or basic?
Basic.
So what are the two forms for the amino group?
NH2 and NH3+.
As we've already seen many times. Now how about the amide? Well, remember, the most important factor in organic chemistry is the charges. And this resonance structure shows us a positive charge. Will this positive charge make this amide more or less basic?
The positive charge? That would make it less basic.
That's right, because remember, bases are electron donors. As you were just saying, bases are electron donors, but if you have a positive charge, you don't want to donate electrons. This explains why amides are not basic. Remember, I was telling you earlier that the amide nitrogens would never protonate, and now we see why. The amide nitrogen is not going to protonate because it already has a positive charge in one of its resonance structures. That makes it less basic.